Hello, this is the fourth video of the respiration topic. It was going to be part of the electron transport chain video, but I realised it was getting quite long. So this is straight after that one really, and it's bringing it all together and seeing how we can get 38 ATP molecules for every one glucose molecule that is oxidised and broken down for energy. So for each one of those glucose molecules, you get 38, and that's like a best case scenario. So all of those things that we talked about before in the electron transport chain, the electrons were dropped off, and then that caused a certain number of protons to be pumped through each time, and then that build up in that chemiosmosis, that build up then meant that they would rush through that enzyme, that weird stalked particle enzyme. They'd rush through that ATP synthetase, and then ATP would be formed at the end of that. So it's not exact, because some of those things, there could be an extra proton that makes it through. They might not be exactly the same every time, but there is roughly a number of how many protons go through each time that equates to, you know, what has been dropped off and what's been moved through. And this is if everything works pretty much as it should do, and the protons move where they should, and it's like a best case scenario, you get 38 ATP. So we'll be able to work out where they all come from. So I'm going to start with the reduced NAD and how that adds up to the ATP first of all. I know you can't really see these very clearly, but that's okay, because you should be fairly familiar with these. I'm only really using to point out whereabouts in the process, so you don't need to be able to see exactly what I'm on about here. And these are the handouts that you'll be given in class as well, so you'll have these when you come in. Remember, in glycolysis, we'll deal with reduced NAD first. There were two NAD that were reduced in glycolysis. Then one that is reduced in the link reaction, this one here. So we've got reduced NAD here and here, and then reduced NAD being formed there. Then in the Krebs cycle, we have reduced NAD being formed when we had NAD, NAD, FAD, NAD. So 1, 2, 3 there. Also remember that because the glycolysis has the glucose at the start, which is then split to 2 pyruvate, after this point, we're looking at here the pathway for 1 pyruvate. So we have that going through this pathway, but we also have this one doing the same thing, so we have to add that in. Okay, so we'll look at that first of all. So NADH, reduced NAD, in glycolysis we form 2. I'm going to do a different colour. So 2 reduced NAD in glycolysis. In the link reaction, you get 1 NAD that is reduced. Remember, the link reaction happens twice, so we get 2. Then in the Krebs cycle, we have NAD, NAD, FAD, NAD. So we have 3 per turn of the Krebs cycle. But if we've come from 2 pyruvate molecules, this has happened twice for every glucose molecule, which means that in the Krebs cycle we have 3 being formed for each pyruvate, which means we multiply that by 2, because there's 2 pyruvate for every glucose, which means that we now have 6 NAD that have been reduced in the Krebs cycle for every glucose molecule. Now if we do the same with FADH2, reduced FAD, we don't meet FAD in glycolysis or the link reaction. First time we come to it is NAD, NAD, FAD, NAD. So we have our FAD here that has been reduced. So on there, oh, I'll put that on there. So it's just here, and it only happens once per cycle. But remember, this cycle is happening once for each pyruvate. There's 2 pyruvate for every glucose. So as the glucose splits and the Krebs cycle happens twice, we have that 1 FAD happening twice. So here we are. None in glycolysis, none in the link reaction, and then in the Krebs cycle we have one, but the Krebs cycle happens twice for every glucose. So we have 2 happening there. Okay. Now, another thing that you don't know yet, but it's just one of those things to sort of learn and accept, is that each NAD that has been reduced, once it gets the electron transport chain, will actually, remember, it drops off its electrons. That causes proton pumps to pump protons, and then that causes this gradient, this chemical gradient. And because of the number of protons that are pumped for every reduced NAD that drops off electrons, okay, so every NAD that's been reduced that drops off electrons causes a certain number of protons to be pumped, and that leads to a certain number of ATP. So you just have to accept that reduced NAD will lead to 3 ATP. So for every NADH that has been formed earlier on, we will later on in the electron transport chain get 3 ATP. And the reason that we get 3, it's all due to the number of protons that are pumped for every NAD that drops off electrons. Okay. And that is another number that can vary. Often it's given as 2.5, and this one will be given as something else as well. And then it will sort of add up to a very similar number later on. But a lot of the textbooks say, and WJEC say, that reduced NAD will equate to 3 ATP being formed later on. FAD, not quite as good. FAD, H2, so for every reduced FAD that goes and drops off its electrons and causes the proton pumps to do their thing, then you get, oh, not much space, 2 ATP. Okay, I have to do that again, 2 ATP. That actually makes sense, doesn't it, though, because in the electron transport chain, FAD doesn't go to the first complex, it goes to the second one, doesn't it? So it misses a proton pump. So it makes sense that there's not as many protons, and then there won't be as many ATP. So that's one of them things that you don't really strictly need to know for WJC, that goes to the second complex. But it does help things make sense later on, when this is all a bit more familiar. So every time you get a reduced FAD that's dropping off electrons, you get 2 ATP. Okay, so what that means is, through glycolysis, the link reaction and Krebs cycle, you get 4, 10, 10 reduced NAD. So if you go through all of this, for every glucose molecule, you'll get 10 NAD. And for every NAD that's been reduced, there'll be protons pumped, the electron transport chain will take place, and you'll get 3 ATP for every one of those NAD that was reduced earlier on. And then that means obviously that you get 30 ATP for all that NAD. So every time an NAD was reduced earlier on, and I was like, oh, it's going to matter later on, it's going to build to the electron transport chain, that is where, that's where it all comes from, really. That's what a lot of the ATP boils down to, is the reduced NAD. Then the FAD, you only get 2 through the whole thing from that Krebs cycle, from your little FAD. They were reduced, they dropped off their electrons at the second complex, they caused protons to be pumped as they moved along, and then that caused chemiosmosis. You got the ATP synthetase that made 2 ATP, so 2 times 2. So you get 4. 4 ATP for each FAD that's been reduced. There. And so we're on 34 ATP, and I said there was going to be 38. But if we go back, here we go, exciting. If you go back, in glycolysis, remember, here we had to spend 2 ATP. Get the thing going, give it some energy. It's like spend money to accumulate some. Later on, you have the ATP that's put in here. Later on, there were 2 ATP formed there and 2 formed there. So in glycolysis, you have 4 ATP formed, 2 spent, so there's a net gain in glycolysis of 2 ATP. Then, in the link reaction, there is 1 ATP. Oh no, sorry, there's not. There's none formed in the link reaction. The Krebs cycle, sorry. When you get into the Krebs cycle, NAD, NAD, FAD, NAD. The was to remind you to stick an ATP in. So it's normally somewhere down here. It varies. Remember, because this is a summary of an even more complicated thing. But down here, you get the ATP that's been formed. And this Krebs cycle, and I've said this a million times, but this Krebs cycle happens twice for every glucose molecule, because it's the pyruvate that goes through this process. And there was 2 pyruvates here. So for every glucose molecule, 2 Krebs cycles, you get 2 ATP there. So 4 formed there, take away those 2, you get 2 from glycolysis, you get 1, 2 from Krebs, so that's where the other 4 come from. So ATP formed just generally through that process. Before you get to the electron transport chain is 4, so we just add all that up and you get at the end 4 up until the point of the electron transport chain. Once you've gone through the electron transport chain and oxidative phosphorylation and ATP synthetase, 38 ATP. Okay. Now if that's too much now, probably is, that's fine. But I think this is one of those things, if you can get to the point where you understand that, then you pretty much understand everything else. So leave this, if that sounds crazy, leave it, go through all the processes again, learn them again later on, go through it in different ways, watch different videos, and then come back to this. And once this makes sense, you'll know that you have everything else pretty solid. So don't worry, because this really is being able to knit everything together. Okay. The only other things I was just going to mention is just a couple of keywords that I have mentioned as I've gone through, but I've not gone into too much because I just want to sort of build it in. So there will be things that I sort of build into this later on, not too much, pretty much there if you're this far. So just a couple of little things. First of all, I mentioned in the electron transport chain that process of the electron transport chain and the ATP synthetase, that is called oxidative phosphorylation. That's because a phosphate, an inorganic phosphate ion, is being added to ATP in an oxidative process. Oxygen is being used. Remember, oxygen was the final electron acceptor. Okay, remember that phrase, oxygen's the final electron acceptor. So that's oxidative phosphorylation. It's a complex process where electrons are moving, protons are moving, oxygen is needed, and in the end, a phosphate is added to ADP, and that forms ATP. That's oxidative phosphorylation. The other type of phosphorylation that you need to know about is substrate level phosphorylation. And this is much more straightforward. All substrate level phosphorylation is where one molecule has got a phosphate, the other molecule then takes that phosphate, it's a substrate. So like when you did about enzymes and you talked about just a general molecule being a substrate and something being added or broken or whatever onto a substrate, that is substrate level. So we call that where a phosphate is added just from one molecule to another, substrate level phosphorylation. But we'll come back to that in school. We'll give you proper definitions for those two, but just so that you've got an idea when you're looking at all the material for all of this, that you get what that's sort of about. The other thing is that you need to be able to talk about dehydrogenation, and I don't think I've mentioned that as we've gone through, but dehydrogenation is just when the reductions, when these are reduced. So when NAD was reduced, so So here and here, here, all those places we've just talked about. Every time NAD is reduced, we said it accepted electrons, it's taking electrons. Reduction is gain, it's gaining electrons. But it's also gaining that hydrogen. So all those reactions we can refer to as dehydrogenation reactions. So when you'll come across this, it might sometimes refer to the oxidation. Let's talk about a specific one. So here in the link reaction, nice and straightforward, remember you have NAD turning into reduced NAD. So if someone was talking about their reaction, they might refer to NAD being reduced. You could also refer to this intermediate being oxidized, and you could also refer to the whole thing as dehydrogenation, because this thing here is losing a hydrogen as well as losing electrons. Okay, so that's what dehydrogenation is. It's every time you go from NAD to NADH, or FAD to FADH, whatever it is that is giving that hydrogen and those electrons is being dehydrogenated. The other term that you come across, which I've probably mentioned as well, is decarboxylation. So here the carbon dioxide was lost. So remember, one of those carbons came off and the carbon dioxide was lost. And here, twice in the Krebs cycle, carbon dioxide is lost as well. So they would be decarboxylation reactions there and there. And that's just because I know now that you're going to go and look at, hopefully, different websites, different videos, and they'll use these words, and they're words that you need to become familiar with. But I'm hoping to layer a bit more of that in later on, because I know it's a lot. Okay.